Okay, we might get started if we can. I think we have our Zoom people joining us now. So welcome to this seminar put on by the Academy of Technology and Engineering. My name is Sandra Kentish and I'm the incoming chair of the Victorian division, taking over from Baldwin Fox, who's done a fantastic job over the last few years. Let me start by acknowledging the traditional owners on the land on which this event is taking place. It's the land of the Wurundjeri people, and I pay their respects to their elders, past and present, and to their families. I would note that this land was never ceded, and Australia was indeed founded on the genocide and dispossession of First Nations people. But getting back to the reason we're here tonight is to discuss a very important topic, which is the translation of research to applications in biotechnology. And we have two great speakers tonight. They're both relatively new fellows of the Academy. And so it's really great to see people in this joining the Academy in this way. We'll start by introducing our first speaker, Professor Sally McArthur. Sally commenced her academic career with postdoctoral studies at the University of Washington in Seattle, and then took up a lecturer position at the University of Sheffield in the UK. She spent the last 14 years at Swinburne Institute of Technology in a variety of roles, including Associate Dean of STEM Research, Director of the Swinburne Innovation Precinct, Director of the Manufacturing Futures Research Institute, and Professor of Bioengineering. But quite recently, she tells me 8 weeks ago, she's moved to Deakin University, where she's now the Director of the Institute for Frontier Materials. So I'll hand over to Sally.
Thanks so much, Sandra. And thanks everybody for being here today. So I'm going to take you on a little bit of a journey through my history of working at the interface between research and industry, and really looking at the human elements of a lot of how we bring people together to collaborate, and different projects and different activities I've tried. And when I put this slide deck together, I sort of went, oh, I think this made sense when I looked at my career, but all of a sudden I've made a story. It's always nice to go back and put things together the right way. So my research innovation journey started when I did my PhD. I actually worked at CSIRO as a research engineer and did my PhD with the CRC for Eye Research and Technology and CBER Vision. So I was enrolled and we were working on the extended wear contact lenses. And so for those who don't know, this was a project where CBER Vision stepped out and said, actually, we need a new type of contact lens that we can leave in our eyes. And they came and CSIRO developed the base material that those contact lenses were made out of that had the right oxygen permeability. They created a new class of materials that would actually work here. Unfortunately, the materials they were made out of are super hydrophobic. So you can imagine putting something that really doesn't like water into your eye is a touch painful. And so the work I did and the work that a large team that I was in at CSIRO was looking at coatings that would go on these contact lenses. Now, I can make you the fanciest coating. I can do everything with that coating, but I can't necessarily make it cost effective to put on a contact lens. And so the challenge became the translation of our ideas for our coatings and how they would work in the eye to a manufacturable product. And that was really my introduction to the challenges of taking research and really translating it into a mass scale up. You can imagine how many contact lenses get made and what sort of coating technologies you can and can't integrate into a contact lens manufacturing process. So this was my introduction, and I was incredibly fortunate to be working with a massive multinational company with a massive team behind it, lots of ophthalmologists up in Sydney who were putting things into eyes and trying to understand what drove the coating to behave in the way it did. Now, when I moved to Sheffield as a lecturer, as a baby academic, in addition to starting up the biomedical engineering program there, which was trying to partner up and understand what did the workforce needs for engineers who were going to work at the interface with engineering, so having lots of chats with companies, we had two spin-outs during that time there. One was called Celtran, and that was taking technology that allowed cells that you take a little bit of biopsy of your own skin, and you take the keratinocytes that make up the outermost layer, and we created a material that would allow those cells to expand. And so 5 days out of needing a skin graft, you could take a small biopsy of skin from somebody's hip or a different area, separate the cells and grow that up in the lab. Now, that technology was the surface chemistry and the materials handling. And so it became a Band-Aid, but a Band-Aid with living cells that would transfer onto non-healing wounds and burns. Now, this project taught me exactly how hard it is to come up with a cell transfer vehicle at the time that you actually need the cells, when they're the patient's own cells. And you think about then, how do you put that into a hospital? How do you get it to the patient when they need it? How do you know how many cells there or what volume of skin they're going to need to replace? And that really taught me a lot about, this was a commercial product. It was commercially available through the hospital NHS system through Yorkshire. And you could actually, they did some beautiful work with this. But that need to go to the next stage of how do you scale it? What's the business model for scaling that product? Which bits of it can be scaled and which bits of it actually have to be done locally as a hospital product at that point as well. And that becomes a different business issue. So in that case, I was seeing exactly how business challenges can take a really great, fully functional product that does a fantastic job of healing non-healing wounds, but it's not clear that you can make it into a product that you can get out into the marketplace. The other end was working on surface coatings for tissue culture polystyrene, things that don't grow, which we like. And this was with the company that was spun out of out of Sheffield as well, called Plasso. And they developed a coating that enabled glucosaminoglycans, GAGs, to be stuck onto tissue culture polystyrene to study how the GAGs and the proteins then interacted. So it wasn't just studying how the GAG worked, the sugar, but it was studying how the sugars and proteins interacted. And traditionally, we didn't have any plasticware that was very good at sticking sugars onto surfaces. So they developed a whole class of materials that could be put onto standard tissue culture polystyrene plates. Again, this was a really interesting exercise where Plasso very early on got venture capital that was run out of America. And suddenly you've lost control over where you're going to go next. And so you're working really hard. You've got a great product. You know that it's going to be really good. You're going to develop it. You know how to scale it. But suddenly, as a team working on this project, all you're doing is trying to chase money. You've lost sight of the science, which got you there, because now all you're trying to do is get money to keep going. So just in this, what I should say at that point, that product, £10 million, I think, was the sale price to Becton Dickinson and Corning of that product. And it was sold on, and it's still made today. It's the Epronex plate that Corning produced today. So these are successful products, but each one of them taught me a different angle on where, as an academic, I wouldn't normally see any of these issues coming up, but it really did teach me a lot about, oh, there's lots of different angles to this. It's not just enough to have a great idea. So I was really fortunate to get a fellowship from the Royal Academy in the UK to come to Australia. I'm Australian, come back to Australia for a bit of a sabbatical after a while. And I actually worked at Minifab, and that's hiding underneath that image on the screen. And so I came to Minifab as my sabbatical. I was based at Swinburne, but I spent 6 months working at Minifab, seeing very early on. So this is 2007. When they were still very much a consultancy firm who did lots of work in the medtech sector. And that, again, gave me a really interesting way of thinking about, well, what are the concerns of a startup that is trying to work as a service provider into a sector? How do we gather together the right people? How do we predict the types of skill sets we're going to need? How do we sell ourselves? And how do we validate that we can actually deliver on what we say we're doing as well? They were doing that really well. So that was a great place to learn how to do it really well. And then as I moved to, I did my sabbatical, said, don't think I'm going to come back to Australia. And 6 months later, I was offered a job in Australia. So, I came back. And at that point, joining at Swinburne, I was asked to pick up the Australian National Fabrication Facility in Victoria, join as the Swinburne representative on that organisation, but also look at what are the needs for biology interfaces in fabrication. And what are the things that the community and the industry would like to be able to do in a piece of infrastructure that we could provide as open access to the rest of the community. And that got me really connected into talking to people about how they might progress their ideas. And that was the starting point for me to really start to think, well, I've got these experiences. What's the next thing to do? How does that come together to do something as an academic that might be a bit different? But also, what else am I learning here? What else can I add that I can bring to this story? And so within the ANFF environment, I built up a bioengineering surface coatings capability that we worked with lots of different people, often just making their surfaces hydrophilic or hydrophobic, nothing too complicated there, but having the ability and working with a team to expose students to those requirements, to being in the university and building up a profile of capability that was reliable, well away from innovative research, but actually finding the right solution, not just a solution to the problems as well. And that's been really good fun. That still runs today. So as we went through this whole environment, I started thinking about, with my colleague Paul Stoddart, there's got to be a better way of thinking about what PhD students can achieve in their PhDs. How can we bring some of the experiences that both Paul and I had up to that point, working with industry and bringing research-led innovation into industrial settings together? And so we went out and started talking to lots of the industry friends we had about town. One of the joys of Victoria is, the biomedical networks that are around town. And so going and actually just sitting down with somebody, buying them a coffee, as is the Victorian way, and saying, so, do you employ postdocs? Do you employ people who have PhDs? Finding out why they didn't, on the most part, employ people with PhDs. What was the barrier? And the fact that in many cases, people felt that they were retraining people once they started to work for them. And so we started developing an idea around a new form of PhD that was really about connecting early career researchers to industry, embedding them in industry from the outset, and creating that opportunity for our industry partners to come into a research environment at the same time. And so how we actually explore the needs of different industry partners, identify the opportunities, and then how these PhDs might develop new platform technologies or new solutions to things that our industry partners had. At the same time, I was asked to head up the innovation precinct at Swinburne. And two of the gentlemen in the front row here, I should say, Board Director Alexander stepped in to chair the board for the bioreactor, and Matthew was my boss as we tried to get the innovation precinct off the ground. And the innovation precinct was about taking it from the other side and saying, as an academic, how do you understand where the opportunities for impact of your research are? How do you get to put a different lens on the work that you do? And how do you get across to people and understand where the match comes? So how do you understand what the commercial reality is? What would be needed of your research? How can you find new opportunities for your research that maybe you'd never thought of before? Basically, how do you go out and have a conversation with somebody? It was a large part of what we were trying to do with the innovation precinct. And so what I thought I'd just tell you about today is a bit about what we actually managed to do in the bioreactor. And so we brought together this fantastic group of small to medium companies around Melbourne and a hospital. And we recruited 10 PhD candidates from across the world. We had 4 women and 6 blokes. We had 5 Australians and 5 internationals. Each student, well, actually, we put them in little teams and they went out and met with They went and spoke to hospitals. They went and spoke to a whole range of different people to identify end user needs in the medtech sector. Keep looking until you have completely exhausted. The 1,000 is sort of a bit arbitrary, 100 each, basically, but it was to make sure that they didn't stop at one idea or two ideas. It was to keep them looking and to look for themes that could come out of those ideas as well, because then you can see the connections and the fundamental technologies and the research that might actually underpin those fundamental technologies. As they brought those ideas back, they came together to really refine them down and to look through the themes in those and to look for the matches to their skill sets, to their diversity of skills. We deliberately chose students who came from a very wide background. It wasn't just STEM. We had social scientists. We also had some medical technologists in the team as well. So we had quite a nice diverse, and one doctor actually as well, one AD doctor. Each of those team members then, having met with all of our industry partners and workshopped ideas to understand their capabilities, pitched different project ideas to the industry partners. So now they're going, well, I've seen this opportunity and I've done a bit of research and I'm now going to give you a 2-minute pitch on the opportunity in this area. So we're training them to get their ideas together, to think about what might be the nubbin of an idea and to pitch it back to the industry partner. And as we went through those pitches, again, there were quite a few of them for each partner, so they could look at a distribution of ideas and go, oh, yeah, that sounds really interesting. Or actually, yeah, we've had an industry partner or somebody's mentioned this to us. Yeah, let's look at this further. And they kept working on those ideas. And so we refined the ideas for each project. So you had to have enough depth of research question to be a PhD, but you had drivers for why you were going to do the research as well, and a really nice focus in. So with the partners and the student, we selected the projects that they would do, and they established project milestones alongside a business case for that project as well. So now they looked at, well, where do we think this could go? And what is the scale of the market? Things like that. Only then did the students work out who their supervisors were going to be. I can tell you now, that broke brains. Because it wasn't about the supervisor's ideas at any point. The relationship was between the student and the industry, and the supervisors came in more as mentors and research coaches to a large extent in the project. And so I supervised 3 projects in this field because the students came to me, had a bit of a chat about it. We had a good fit, but it also taught me that I could lead research or be part of a research team without being the expert. I knew how to do research though. And that in itself, I knew the processes to follow. But so much of the time, what we were looking at is what is possible with the boundary conditions we have. If we can't completely redesign the product because it's going to be too expensive, we have to think about the analysis techniques that'll give us insight and value for what we can change. And so again, it's proper industry-led research at that point. It's very focused and you're making choices based on not the plethora of all the ways you could go, but on that there is a specific question that we think we have to answer. So let's look at how we answer it. And that's going to help us move forward. Each student also had an external mentor, and I'm not going to tell you that this was an easy process, okay? This first stage took us 9 months to work through with these students, okay? And there was a lot of nerves. We actually brought in a psychologist to work with them as a communications psychologist and as a team psychologist to help them work through. What we also did through this process was actually get them to really think about how to work in a team and how to communicate their ideas. So by the end of this, they were really very good at bringing ideas together quickly and working out what the opportunities might be as well. So out of this, we completed 8 PhDs and 2 masters. There were 6 patents generated, 3 new products that are in the market as a result. We've got employment in industry. We've got employment in local industry. We've got a biofuel startup that's come out of it as well. We've got academic roles that have come out of it as well. There was funding obtained to do the next stages of a product development within the academic research. And as I said, this new model of PhD, which is PhD in technology innovation, was actually built. And this model has now been translated at Swinburne into the impact PhD and looking for impact. So it's a cohort model. So it's designed to bring a cohort of students together, working together to explore spaces as well. It was fun. Can I just say that? I really, really enjoyed doing all the work on this within this. And you never quite knew where you were going, but you knew when you got there that you'd done the right thing. And that was great. And these people who are the students who have come out of these, I fabulous, got to say. But I also learned some stuff along the way here, which was in many of the companies that I've been talking to, and as we continued to talk through the whole process, there was so little research capacity. Business as usual didn't have capacity to do extra research within the business model. And in many cases, they've never done research. They didn't know what research might do for their business either. Not in our partners, they'd obviously joined us because they understood it. But as we kept talking to more and more companies around town, they had very little idea on what problems might be able to be solved by partnering with universities. And obviously, it's all about trust. This was a risk that all of these partners took with us to come in and be part of our centre. And so finding those right partners was the work that we did in actually establishing what is the right type of model for a PhD for you. And it takes time and it always needs help as well. So it's those relationships. And it was the same in the work that we did in the innovation precinct. Getting academics to trust you that you weren't going to try and turn them into a business specialist. You know, the number of times somebody said, oh, I know nothing about business. And I'm like, yeah, it's not about that. It's about asking really good questions. It's about mentoring by saying, well, have you thought about it? Not knowing the answer, but learning with them as well. So this is where when Karen Reynolds from Flinders University approached me about working on the medical device partnering program and bringing it from the University of South Australia into Victoria, I got really excited because so much of what I'd seen coming out of the bioreactor played into the need to build these communities that had diverse groups of people who could comment on an idea, who could give feedback, and who would be needed along the product development journey as well. And bringing them together early in the idea, not running into them later on and going, oh, I wish I'd met you 5 years ago. You could have really helped. And so for those who don't know, Karen built up this environment in South Australia where she brings together hospitals, medical device companies, end users, research organisations, service providers and manufacturers. So you've got all the ecosystem coming together and they all work on different ideas. I'm going to have trouble getting through this. And so this is what we did in Victoria. With the support of Launch Vic, to the tune of around $2 million, we built up a partnership across all the universities, except Deakin. We brought together CSIRO, many of the research institutes, and then the patent people as well, and different manufacturing and design houses as well. And we take everything through this process where anybody in Victoria could give us, who was wanting to develop a new medical product, could come in and say, I've got this idea. This is the evidence I've got for why it's a good idea. And we would assess it. So we would screen it and we would look to its clinical need and we would go out and talk to people and say, so we go and talk to clinicians, we wouldn't be disclosing, but is this a problem for you? Those basic questions. And then if we got positive feedback in the clinical environment, we would say, okay, would you be interested to come to a workshop with us to help work out how this product might get to market, where it might go, where it might fit, what technologies might be needed to make it real. And so we would run a workshop for a couple of hours with a very fixed set of questions that we went through, understanding the clinical need, understanding the technical need, and understanding the road to implementation, and trying to bring that community very early together around these ideas. At that point, we were looking to see if there was a small research project that we could fund that would help solve a technical problem. And that was our limitation here. We were looking for something that was going to be R&D, that a university or one of our partners could deliver with that company to help them get over a small problem or a big problem that they might have on their journey. And so that was where we funded a project for 3 to 6 months in a university or a research provider to develop a solution and do a handover to that company. Now, through that process, we looked at, in 2 years, we looked at 130 ideas. We ran 47 workshops with over 250 participants in those workshops. We created 17 new prototypes or validated 17 different new products. And we created about 65 jobs. Now, that was just in that 2-year period from zero in Victoria. We had no ideas being generated into Flinders or South Australia before that, and just gathering these ideas, evaluating them. A lot of them were great ideas, but our criteria required that there was a novelty. So we could then connect everybody at this 130 ideas, got feedback, got connections, and got connected to different communities. One of the most valuable things I think we found along this journey was the clinical insight and how hard it was to get clinical insight into your products early in the product development life cycle. And what we found also was that this area of assistive technology kept coming up again and again and again. So the last thing I'm going to feed into is then how that changed what I thought about when we were putting together MedTech Vic at Swinburne University with the support of the Victorian State Government. And we brought together a range of assistive technology firms to look at manufacturing and development of assistive technologies, but in this instance, co-created and co-evaluated by the end user. So this is people with the need and their caring community. These products were going to be useful and usable, accessible and adjustable. They need to be manufacturable, bringing back all of these pieces that I'm hoping is making sense. Really important one, desirable. Something that somebody actually wants, not just the solution that they have to carry around with them, but the thing they actually would like to, aren't too worried about having with them. And we'd really like them to be locally made. So we developed up a co-creation space, which has just opened at Swinburne University, that creates a home base for everybody to come together that's accessible. And we put a lot of effort into the accessibility in the space. But that's about bringing community together and diverse community together and really thinking about how to do that. And then we created also the Clinical Innovation Fellows program, because again, after all of that clinical insight, we realized that actually there were a lot of clinicians out there who wanted to know more about product development. We've now run 2 cycles of this program and trained up 14 clinicians from across Monash Health and St Vincent's in product development. So we've given them the basics of how does a product go from an idea all the way through, so that they can coach, mentor, participate in those early stages of product development. And we're now developing a lived experience fellows program. This is for people with disability to learn how to be coaches and mentors into companies developing new products and services as well. So this is me now. I'm at Deakin, and I'm now trying to bring this all together around a completely new area, looking at materials for low carbon living. And so this is energy, this is all of how our future is going to work, and sustainability. So we've got a lot of new work to do there. Before I get the hook around my neck and dragged out, I just wanted to close with how it takes a village to get any of this to work. And it's all about the people. It's about building the trust. It's about sharing the vision, sharing knowledge, sharing outcomes. And I think this is the thing that is really important, sharing the wins, the losses, and the draws. So knowing when to walk away and when to run. I will break into song if you let me. But also sharing the risk and finding those partnerships of understanding the level of risk that somebody is willing to take here too. And really coming back to all the people who can lead and all the ways that we can lead in research translation, but really human-led being at the heart of everything we need to do. So thank you.
Thank you, Sally, for an excellent presentation. We have run out of time.
I know.
So we won't allow any questions at the moment, but if we have some time towards the end, we might be able to sneak a few in then. But I'm now going to pass over to our second speaker, Dr. Andrew Nash. So Andrew completed his PhD in immunology at the University of Melbourne in 1988 and developed and led a research team focused on basic and applied aspects of cytokine biology. He then joined Zenith Therapeutics, which was then AMRAD, as a senior scientist, and subsequently held a number of positions, including Director of Research and Chief Scientific Officer. In July 2005, he was appointed CEO of Zenith, a position which he held up until the acquisition of Zenith by CSL in November 2006. And following the acquisition, he was appointed as CSL's Senior Vice President Research. He's currently based at the Bio21 Institute, where he leads a large global effort focused on the discovery and development of new protein, cell and gene-based medicines. So I'll pass over to Andrew. Thank you.
And what a great presentation to follow on from, Sally. Really enjoyed that. It's a pity we didn't get a chance to ask questions because I had some, and I'm sure plenty of others did as well. But I guess, you were talking about creating an ecosystem in which translation can really flourish, and those academics, those, inventive people that want to come up with ideas and translate into practical use, are able to do that and find people that encourage that. And, your focus is on devices and technologies. What we've been thinking about is very similar, about the ecosystem that will enable the development and translation of new therapies from a precinct like Parkville, or in fact, anywhere really. So what I'm going to talk about today, you may have to talk a little bit about CSL R&D just to give you some background as to, where we're coming from and our interest in this space. But then I'll talk about what one of my real passions, and that's the translation of early research through into novel therapies. What are some of the challenges that I've seen over the last 20 or 30 years of research here in Melbourne, and some of the opportunities I think are really moving forward at a rapid pace now. And I'll talk about some of the CSL and partner investments into that space to try and address some of those challenges and realise some of those opportunities. I'm talking about CSL investments because they're the things I know about, but obviously a lot of others are interested in this space as well and investing in much the same way as we are. But just in terms of CSL research and development, it has changed a lot over the last, probably 15 to 20 years. From a company that was primarily technology driven, we were able to collect plasma, we were able to fractionate plasma to medicines. So the therapeutic areas we're interested in really related to what it was we could get out of plasma. And of course, we made influenza vaccine as well. And that's really transitioned over the last decade to a company that has specific therapeutic area focus and strategies that are aligned to those therapeutic areas. So now we think of ourselves as a company with drugs and R&D activities in the immunology space, hematology, respiratory transplant, and cardiovascular, as well as vaccines, and particularly influenza vaccine. And our activities across those therapeutic areas are supported by really 4 main drug discovery platforms. Of course, plasma is still a big interest to us. What new drugs can we get out of plasma? Recombinant technology has been a very important part of what we're doing. We're dipping our toe in the water of cell and gene therapy. We've got clinical programs ongoing, and hopefully we'll have a product approved shortly. And of course, we've got our flu platform, which is evolving very rapidly at the moment as well. In terms of the effort in R&D, it's really developed over the time I've been at CSL. I've been, as was said, at CSL for probably about 15 or 16 years now. When I arrived there, we were spending around $100 million a year on R&D. And last year, and that was Australian dollars, last year we spent 1.2 billion on US on R&D. And we have about 2,500 people across the globe in R&D. In early stage research, which I look after, our main focus of activity is here in Melbourne, because Melbourne's such a great place to do early research and to look for opportunities that can be translated. But we have big groups in Switzerland and in Germany and in the US in Pasadena as well. The investment is across the whole pipeline of R&D. So we invest probably 10 to 15% of our R&D dollars in early stage research, and then the rest goes towards product development, subsequently clinical development, and then regulatory approval as well. So we've really transitioned from a company that developed a lot of technology, looked to partner and took some things forward itself, to a company that whose R&D effort is directed towards products that we can develop and move forward ourselves. Yet occasionally we still look at partnerships for obvious reasons. Now, I just want to give you one example of some of the work we do and show you a little bit of science, because again, that gives you a sense of the capability we have in CSL and the things that we're really interested in. So we have been a company that for a long time has worked in rare disease. So it's a very important area for us. Hereditary angioedema is one of those rare diseases, autosomal dominant genetic condition, around 1 in sort of 25,000 or so people. So pretty rare. You can see what happens in this genetic condition over there on the left-hand side of the slide. It's really spontaneous, uncontrolled swelling and edema. And depending on the location of that swelling and edema, it can either be just very, very painful or it can be fatal. So it has been largely unrecognized and relatively poorly treated for quite some time. It's all about the lack of a particular serpin, C1 inhibitor, which regulates factor 12 and the bradykinin and kallikrein pathway. So these mediators are what leads to vascular permeability, swelling and edema. Upstream, this pathway is activated by factor 12. And in these people with its genetic deficiency, they're missing the major regulator of the pathway, C1. And for a long time, C1 from plasma has been the mainstay of therapy. It's problematic. It's hard to purify. It has a short half-life, and it's really hard to treat these people prophylactically. So generally, they only were treated when they had an attack, and that was often too late. So we looked at this pathway. We did a lot of really early basic research here in Melbourne and in our labs in Marburg, understanding the role of factor 12. And we chose to develop a therapeutic antibody that targeted factor 12 to antagonize and neutralize that activity. Most of that work was done in Melbourne, the development of the libraries that yielded the antibody, the development of the clones, the manufacture of the antibody, the early clinical programs were done here. And these ultimately led to the phase 2 study that you can see here, which was very successful and recently published in Lancet, where these were a group of patients that were on average having around 4 or 5 of these attacks per month. So every week they're going through that sort of experience, to when treated with the therapy that we developed here, really having no attacks at all. So a very successful phase 2 study. This program has just finished phase 3. All the primary and secondary endpoints were met, safe and well tolerated, and we're hoping to file and have the drug approved by mid-23. And this will be a real improvement in the treatment of these patients. So this is really an example of our work in the immunology space, where we've been able to take our capabilities in basic biology and science, understand how this particular pathway was contributing to the pathology that was observed, identify the best target, develop the drug, test it in animal models, go all the way through phase 1, phase 2, and 3, and end up with the product. So this is the sort of thing that we're really interested in doing, and we're really interested in creating an ecosystem, an environment that really encourages this type of drug development, encourages our scientists and academic scientists to really think about illness, untreated disease, and what are the best ways to address that. Now, I'm going to just move on to some of the things that we see as issues and challenges in translation in the drug development space, and some of the things that we've been trying to do about it. So from my perspective, and I've been working in the Parkville precinct for a long time, and I've seen this across other medical research precincts across Australia, is that there was really quite a big disconnect between scientists, industry, and clinicians in hospitals. I'd say for the last 25 years, you would struggle to get the really great scientists in Weehai doing much work at all with some of the clinicians in the Royal Melbourne Hospital and other locations. And there was very little industry involvement. And in fact, a lot of the academics weren't interested in translation in industry. And it was really hard to get clinical work or translational science done within the hospital environment as well. And that led to this sort of winding pathway of drug translation where it was very haphazard and almost good luck that you ended up with a drug at the end. So I think this has really changed a lot over the last 10 or 15 years. It's changed hugely. There's been a lot of investment, and I think there's a lot of investment to go. But I think this is where we want to end up, where these three disparate groups that are really key to the translational process are working much more closely together. So we want globally competitive funding for basic research. We want clinician researchers that are really dedicated to the development of new therapies. And we want industry which is co-located within those precincts and has the capacity to do the translational science and research that can lead to the drug discovery process. And when I was putting this together, I was thinking about, well, what do these overlaps represent? And that little blue piece there, I think, is the overlap that represents a truly productive medical research precinct. So within that overlap are all the things that we need to happen for early research to translate into outcomes. The circle that I put there is, the red circle is what I think of as, a biotech incubator space where industry is working very closely with academics. And I'm going to talk a little bit more about an incubator that we're constructing at the moment. But in terms of making all of that work much better, in terms of going from the past to a successful future, what are some of the things that have needed to happen? And what are some of the investments that we and others have made to really develop that ecosystem in a much more productive way? So these are some of the things that we're involved in at the moment. So certainly more investment is critical. We're very strong supporters and advocates for more investment into early stage medical research. We love what MRFF is doing, but we're very disappointed with the way NHMRC has been travelling over the last decade or so. It just hasn't been growing at the rate that it should have been. And we're in there advocating for increasing investment all the time. We're participating in the venture capital space. We're investing with venture capital firms. We're investing in accelerators and those sorts of environments. And we're also now directly investing in startups. So where technology or things that we've developed don't really work for our portfolio, and that happens probably more than you would think, we're looking to spin out that technology and partner with venture capitals. We're also looking to create more investable opportunities. There does still seem to be a gap between what industry understands as investable and where there is a clear path forward and where the science might be within the university, the academic sector. So we're working with our colleagues to try and create those investable opportunities. We set up last year the WEHI CSL Centre for Biologic Therapies because CSL was very strong in biologics. WEHI, great outcomes in small molecules, but really no biologics capacity at all. So we moved all of our technology across into WEHI for them to make use of it. We've got a research acceleration initiative going where we're out funding research, early stage ideas. And what I really want to talk about is the effort we've made towards developing the innovation ecosystem, particularly here in Parkville, but also elsewhere in Australia. It's about people, it's about skills, and it's about infrastructure and technology. Our research group is based here in the precinct. It's our global hub. We have 150 scientists working at Bio21. We employ 130 PhDs at that site doing drug development. And unfortunately, that's by far the biggest employment of postdocs in medical research in Australia, but it's dwarfed by what happens overseas. And we have a Biotech Incubator partnership that I'll get to. And we're also building some of the GXP capabilities that are required for drug development. Now, just to look at some of these and walk you through some of the examples, all of this is built off a very strategic approach to external innovation. So around 5 years ago, within the CSL Research, we set up an external innovation team specifically to look for ways to engage effectively with the research community. That team is based here in Melbourne. There's quite a few people working for that team here in Melbourne, but they have also people in Europe and the US. And this is the model they use to guide their efforts. So it's about funding and collaboration. It's about partnership with incubators and venture people. It's about making use and leveraging the sites where our researchers are based, because we often find that collaborating nearby and having our scientists directly involved with scientists within the institutes is the most effective way forward. Long-term strategic partnerships, as well as shorter-term project-specific objectives. And I think, of course, scouting and looking for disruptive therapies. And there's a few examples here. So we have long-term strategic partnerships with WEHI, University of Melbourne, Seattle Children's Research Institute, University of Zurich, just to name a few. In terms of incubators, we're a big investor in Brandon Capital. I'll talk a little bit more about that shortly, but also a number of other accelerators and VC groups in Europe. And, you know, this all comes together for a very strategic approach to our external engagement. This is led by Martha Don-Brain in my group, who's an outstanding scientist and business person as well. So this is the outcome of our research acceleration initiative. So we're trying to capture here 5 years of work where we've been out in the Australian community in Europe looking for really early stage ideas and investing in them for about a 2-year period, trying to create investable assets, trying to generate projects where the probability of success is improved because of our involvement, and we can bring them into our portfolio. So we're looking for novel targets, novel therapies, new platforms, and a lot of these are based on direct engagement with CSL scientists. And it's been a very successful program. You'll recognise a number of the institutes down in Australia that we're funding this work in, but also a number of really key institutes in Europe and in the US. And just in this year, we've been successful in establishing really important relationships in Singapore and in countries in Europe that we haven't previously been into as well. So we use this to generate a portfolio of early stage projects. And currently there's about 35 or so projects within this early portfolio. So this is CSL out investing in the academic sector, helping create opportunities that can have a better chance of successfully translating. This is some of the investment we're doing in accelerators and with VC groups. I'll just focus down here in Australia. So Brandon Biocatalyst is the biggest venture capital fund in Australia in medical drug development by far. We're about a 10% shareholder in that fund, along with 4 big superannuation funds. So there's us and the 4 other big guys. We own about 10% of the fund, but it's a fantastic investment for us in order to see what's happening across the broader Australian sector, but also to spin off some of our ideas. And one really nice example is a spin-off company we have here in Melbourne with the dental school, where we're developing a vaccine to treat periodontal disease. Been going very well, and we're hoping clinical studies will start next year. We own about 30% of that company. I think the university owns 10% and the VC guys own the rest, but it's a great collaboration. Just in terms of our presence here and what we're doing with infrastructure, this is our facility down at Bio21. It's our global hub for research. So this is where most of our scientists are based. This is where we make the biggest investment. There's 150 scientists here. There's 4,000 meters squared of wet lab space, and a whole range of groups that you would expect you would need to develop early stage research. So scientists doing science that you will all be familiar with. The science we're doing on the bench at the very early level is no different from the science that's been done within WEHI or the University of Melbourne or Monash. It's just that it comes from a perspective of therapeutic development rather than basic biology. But the quality of the translational science, the quality of the data science, the quality of the analytical biochemistry is all first class with some really outstanding scientists. The other thing that's happening at the moment, and you might have noticed it when you're on your way here today, is the big building just across the road there on Elizabeth Street, which is to be finished, I think, in the first quarter of next year and will be occupied by CSL. So this is a really exciting move for us. The big site up in Poplar Road will shut down in about 2026. And everyone will move down to this site to be closer to us at Bio21. And we like to think that they're moving down here because the presence at Bio21 has been such a success for research that the rest of the company has followed us down here. And while that's not completely true, there's certainly an element of truth in it. So 800 employees across about 15 or so floors here, office space. The really exciting thing is this is not an office tower. This is a research facility. So yes, there's 7 floors of office, but the other 10 floors are GMP product manufacture, research labs, technical development lab, lots of scientists doing science in this building. So, you know, it's making great progress. I had a tour the other day. It's coming along very nicely. But from my perspective, the really exciting thing is that across 2 floors of this building, we're partnered with Melbourne University and the Hall Institute to create a biotech incubator. So this is a $95 million project, housed across 2 floors. It's not going to be run by CSL or the University of Weehite. It's going to be run by an experienced incubator operator, and that operator is going to be announced next week. It's got lab space, office space, and it'll house up to about 40 startups. So we're really keen to get small companies in here, ranging from 1 or 2 people through to 20, 30, 40 people. This is all about the ecosystem. These companies don't have to be in the CSL space, and CSL doesn't have any preemptive rights to what's coming out of them. It's all about an investment in the future of the ecosystem here in Melbourne. It'll launch in early 24. Hopefully we're bringing that forward, but I think it'll be a really important step for the Parkville precinct in terms of having this facility. It's been really well supported by Breakthrough Victoria. This was the first investment they announced. Breakthrough Victoria has a lot of dollars, and I think they're investing them pretty well. They're investing in early companies, and they want to see an environment where those companies can flourish. So an important investment for them was the creation of an incubator that looked like this. So just moving quickly to some of the other facilities. This is what is happening out at Broadmeadows, or it has happened out at Broadmeadows over the last 20 or so years. This has been a huge expansion in advanced manufacturing in Victoria, starting from the very early establishment of the plasma fractionation facility in 1994, all the way through to what you can see up here in 6, which is a global facility for the base fractionation of plasma for the development of or the production of products that will go all over the world. So a lot of plasma coming from Europe and the US will come here into Melbourne, be fractionated in this facility, and then go back out to the rest of the world as medicines. You know, perhaps one of the most exciting things for me is this facility here, which is our phase 3 launch scale recombinant manufacturing facility. This is the facility that manufactured the garadacimab for the clinical program I showed you earlier, phase 2 and phase 3, and that it's producing material for launch. But it's also the facility that made 50 million doses of AstraZeneca vaccine. And, you know, while AstraZeneca vaccine got a bit of a bad rap, most of that was unwarranted. And it was the vaccine that protected Australia in the first instance. And we were really pleased to be involved in that. So a great site with some really advanced manufacturing. And this is probably the most current and biggest investment in Melbourne's manufacturing capability and R&D that we're making at the moment. And this is a new flu vaccine manufacturing facility being built out at Tullamarine. And this is really a major advance. It's taking flu vaccine manufacture from the egg-based process that McFarlane and Burnett came up with in the 1940s, and which is still used to produce 95% of flu vaccines today, and transitioning that to cell-based manufacture, which CSL has really led globally in with the acquisition of a business in North Carolina. So it's not long in the future that not many flu vaccines will be coming out of eggs. Most of them will be coming out of facilities like this. So this is a really big investment for us as well. So just to finish up in terms of, you know, translation and innovation and its importance in Australia, I think, you know, there's been a lot of progress in Australia in the last 20, 30 years in terms of creating that ecosystem. It's clear we still lag. If you compare how we're progressing towards where the US is and where we're seeing Europe move to, we're still a long way behind in terms of having the environment and the right mindset to translate our research, but I think we're really getting there. Skilled workforce is a real issue for us. I mean, it's a big issue globally at the moment, recruiting and retaining people, but it's particularly tough here. This is something that annoys me no end, that we really need a national collaborative framework when it comes to state and federal governments in infrastructure investment. I just cry every time I see Victoria, New South Wales and Queensland competing for the same infrastructure and the whole thing going down the toilet. But, we'll get there sooner or later. And when we do, and when we continue down this path, I'm sure, there's lots of opportunities for translation of medical research here in Victoria. And that's it. Thank you.
Thank you, Andrew, for a fascinating presentation, and you have finished in time to allow us a few questions. So I will open the floor to questions for Andrew and also potentially for Sally if there is anybody else who wants to quiz her as well. So open the floor for questions.
Perhaps I can start asking Sally a question.
Okay, you can do that. Go for it.
I thought your model was fantastic, Sally, and I was wondering, what is the level of investment you're getting from your industry partners back into those PhD programs? Are they putting skin in the game, or are they sitting back and capitalising on your hard work?
That first cohort was actually funded through a training centre, so an ARC training centre. So each industry partner paid the costs of the project itself. So that ran at about $30,000 a year. So it was about equivalent to the scholarship.
Yeah.
So, it was a pretty good split. We have run since then a number of cohorts of students that we've taken to put into that model with an industry partner. It's still at that very early stage of really finding the right partners who want to do a cohort as well, because it really does, it's in isolation, it's not as effective as having a cohort of diverse students together. So we've been trying a few different ways of doing it. And I'm going to try and do a bit more at Deakin. So it's coming with me.
Great presentations, and for someone who's been around the field, it's great to see the new environment. But I'm conscious that our universities have been fairly inflexible how they want to work with industry. Sally, how do you fund students for a year and then pre-PhD? So what could the universities do more of to stimulate the environment you guys have created?
Well, I can give you sort of a really near-term and relevant example, Paul. The MRFF funded the Ready Fellowship Program, and we've been involved with that with Melbourne MTP Connect. And at our first go at CSL at finding people to come into CSL on those fellowships, we had great candidates lined up, but to get released from the university or to have their NHMRC funding or ASC funding paused for a while in a way that would enable them was impossible. And so one thing that could happen would be to create the flexibility that enabled exchange from universities into industry and vice versa. And actually that has noticeably improved over the last couple of years. So the last rounds of Ready, I think we've found, more willing partners, I think also with the cutbacks in the universities, we've actually been very careful about strategic funding for some of these things. And I know at least as part of what we're doing and looking to take the model actually of CSIRO's CERC postdocs and bed them more into things in their postdoc. So making sure that postdocs get more exposure so that you're not locking a postdoc into only a project during that period, that there is more focus on what else are you going to do in your career. That's a real career development stage. So I'm looking to see how we do that with the Alfred Deakin fellowships at Deakin University. They're the ones we fund ourselves. So if we can extend them by going into partnership with industry, that's a great way of thinking about them and having a year in industry as an extension of those types of things. I think also the internship, the push towards internships for PhD students helps. But I also think fundamentally it's creating more opportunities for our undergraduates to do these experiences and get research experience in their undergraduate degree so that they can come through these cycles as well. Because if you come through having done some time at CSL as an undergraduate and then know that what a PhD could, that's how I did my PhD. I worked at CSIRO, worked out that a PhD was actually what I needed. And so went to a PhD having had some time in that sort of industry environment. Yeah, I think that's right. I mean, we've got a fantastic data science group, a group of bioinformaticians, and we built that out of the AMSI intern program.
Yeah.
So that was taking mathematics, computer science PhDs, bringing them into CSL, and using them to create our own team. And now that group has grown to like 20 or 30 people and is recruiting from all over the place.
And one of my postdocs works for it.
Right, there you go.
I think we have time for one more short question. If we, no, you, I knew you had your hand up before, yeah.
Both of you really, and thanks for the presentation, I enjoyed that. But one of the problems I have in the past is that, you know, you want to do a PhD student in and work in an industrial project. But as we subtly know, most projects don't work actually. And you always have this problem of saying, well, a year and a half later, we don't want to do that anymore. Actually deal with this with PhD students. I mean, how do you it strikes me as a different situation.
Well, I mean, we often co-supervise rather than have the PhD student fully embedded. So it'll be a basic biology question where, you know, they're partnered with an academic group and, you know, the PhD will follow the normal course of an academic PhD, but with a therapy in mind. So that's one way to deal with it. We don't have too many PhD students here in Australia that are solely within CSL doing a PhD. We do in Germany though, and the system's a little bit different there.
And I think that's the other thing. There is no rule that says a PhD has to be one project. As long as they're moving you up the knowledge level, my PhD started in complement activation. It did not end in complement activation. Complement was not mentioned and it changed at 18 months. So I think we get a bit fixated on what if it's going to fail rather than how about we just have a go at it and we'll deal with the problem when it happens. Because I think we can be really creative along that way, that there is still knowledge generated in that first 18 months of you'll identify what the next stage is. There's no guarantee in any PhD that it's going to work. You know, it's what you get really, and it's what you choose to do with it and how you demonstrate the learning and the knowledge at the right level. I think we think our review community is actually really rigid, and I don't think they have to be.
But we should end there because we have run over time. I'm conscious that Zoom might leave us in a minute. But can we really give a round of applause for our speakers? And if I can just finish in closing by letting you know about our next event, which will be on November 3rd. And we'll focus on challenges in the agricultural and food sector. We'll be delighted to have as our speakers three of our most younger people who've won the ICM Agri-Foods Awards in the recent past, but it'll be chaired by Professor Paul Wood. And I would like to acknowledge just in closing, I believe he won an award recently as the Eureka Mentor of the Year. So it will also be great to have Paul chair the event next time. So if we continue to